This Week in Virology, the podcast about viruses, the kind that make you sick. This Week in Virology, episode number 258, recorded on November 8th, 2013. Hi, everybody. I'm Vincent Racaniello, and you are listening to TWiV, the podcast all about viruses. Joining me today from Ann Arbor, Michigan, Kathy Spindler.
Hi, Vincent. How are you?
I'm well. How is Ann Arbor? Is it warm and sunny?
It's definitely sunny. It's about 7 degrees. And what is that in Fahrenheit? 44.
Chilly. No snow, though, right?
No snow. But they were talking about potentially snow overnight, but it wouldn't stick, So
I'm heading out to Madison on Tuesday, so I guess it'll be chillier than I'm used to here.
Yeah.
We have clouds in the sky today, 9 degrees. Not such a great day. Also joining us today from Western Massachusetts, Alan Dove.
Good to be here.
How are you, Alan?
Doing okay.
Excellent.
Weather here is a whole lot better than the weather in the Philippines. I have no complaints.
What's going on there?
Oh, biggest typhoon ever recorded, I think. Just made landfall there yesterday.
Oh, that sucks.
100 and some odd, 120 or mile an hour, some ridiculous wind speed.
200 something, I think I heard.
Wow. Oh my gosh.
Yeah, it was huge, yeah.
It's terrible.
Huge. It's going to be a massive mess. Anyway, it's, partly cloudy, even some little smatterings of rain today. Not too bad.
You keeping up with your fallen leaves?
Yeah, I mow them.
Really?
Good.
I have a mulching lawnmower. Most lawnmowers these days are mulching.
Yeah.
And for the past several years, my way of dealing with the leaves is to mow them.
But don't you get tons and tons of ground up leaves in your grass?
And the grass eats them in the spring.
It's no problem, yeah.
It works beautifully. There was some research done on this back in the 1990s at a couple of agricultural schools that have turf management programs. And they found that they could put huge amounts of leaves on the turf and just mow them in with 2 passes of a lawnmower, and everything was fine.
I have to try that this weekend because I've been blowing mine away.
The whole leaf removal thing is a total scam.
I blow them into the woods where they then make nice dirt and they get great topsoil.
Yeah, that's fine
But,
if you have convenient woods to blow them into.
I do. So I've been using gasoline from Sandy a year ago that I had left over from the generator, and it broke my blower, of course.
Oh.
I brought it in to get fixed, and the woman said, "Are you using old gas?" I said, "Yeah." That was it. I had to clean it out. We have a special guest today. He hails from the fair city of Baltimore, Maryland, and before I introduced him, can anyone guess who he might be out there? He's been on TWiV more than any other guest. And if you guessed Matt Frieman, you'd be right. Welcome back.
Thank you very much, guys. Happy to be here.
Coronavirus expert, right?
I do work on coronaviruses.
I know you don't like it when we say that, but you are we don't say you are the most knowledgeable expert. You're just a expert, right?
Yes, I'll go there.
Because we don't want your coronavirus colleagues to give you hell.
Right. I probably know more about coronaviruses than 99.9% of the population in the world. We'll go there.
You know more than we do.
Mm-hmm.
That's for sure. And that's why you're here today, because we had a cool paper come out recently, and we thought we'd get you on. So we appreciate your joining us.
Sure.
Hey, what's the weather like in Baltimore?
It's actually very pretty today. Nice blue sky. I think it's about 50 degrees.
Wow.
It's a nice fall day.
That's pretty nice.
Yeah.
Much nicer than here. I passed through on the train on Monday. I go to ASM a lot in Washington. And I always pass through Baltimore. You can tell all those row houses. It's amazing in that one area. And then, of course, Hopkins towers above everything.
Right. You see, yeah, they actually have 2 new big towers they just built, opened last year. So on that side of the city, that's the main attraction.
That side. I always think of you when I pass through Baltimore.
Thank you very much.
Matt Frieman. Before we do some science, we have a follow-up from Joe, who writes, "Dear TWiV cats, I enjoyed your lively discussion on the veracity or not of scientific research and wanted to chip in. I think it's important to recognize the limitations placed on science by technology. Our observations are only as good as the instruments we use to make them. For example, to our distant forebears, it made intuitive sense that the sun revolves around the earth. It seems true. We can, after all, see it with our own eyes. It wasn't until Galileo used his telescope to track the moons of Jupiter that the true nature of the solar system was accepted. I use advanced microscopy to study viruses, and this is another good example. Using standard fluorescence microscopy, a virus could be seen to co-localize with a cellular protein, and you may interpret that they are in the same place or even interacting. However, in this case, the resolution of the instrument is limited by the diffraction of light. Advanced techniques such as super-resolution fluorescent microscopy or electron microscopy can see beyond this limit and may reveal that although the virus and protein are in the same cellular compartment, they are very much distinct. It is not so much that the original observation is false, more that the high-resolution observation is more true. In this way, advances in technology may allow us to incrementally refine our truths, although it is arguable whether this process could ever be brought to conclusion, allowing us to reach a solid gold truth." As an aside, I'd like to throw in this quote from scientific deity Richard Feynman. "I have found out how hard it is to get to really know something, how careful you have to be about checking the experiments, how easy it is to make mistakes and fool yourself. I know what it means to know something. I suggest we all think what would Feynman do, and science will be just fine. Cheers, Joe from London." It's a good point, right?
Mm-hmm.
I partly agree. New technology gives you better precision, but in many cases, the fundamental insight precedes the more precise technology. I mean, the Earth revolving around the sun is a perfect example. It wasn't Galileo; it was Copernicus who worked that out based just on observations of the movements of the planets and the stars and the sun.
Mm.
And it made more sense mathematically that the Earth revolved around the sun rather than the other way around. And Galileo's data on the moons of Jupiter that he got with the telescope certainly bolstered the argument. But Copernicus was there first.
I guess in some cases the technology will change conclusions, but I have a feeling it's not probably doesn't account for all the perceptions of science being wrong, right? It may be a little bit of it, a fraction of it, but
yeah, and certainly new technology drives a lot of discoveries in science. You know, if you look at something like the microbiome or the RNAi phenomenon, that's been hugely both of those have been hugely driven by new technologies that we have to study those things. Yeah.
We have a SARS paper this week, and that's why Matt is here. This is a paper that was published, a letter in Nature. Isolation and characterization of a bat SARS-like coronavirus that uses the ACE2 receptor. And this is from a very large group in China and also the Emerging Infectious Diseases Program in Singapore, Australia, University of California, Davis, the EcoHealth Alliance, which is right here in New York City. Wow, how about that? They got a new SARS-like virus right here. No. But anyway, there are lots of authors. The first author is Ying Yi Gay, or G-E-J, or Ge, I don't know how to say it. And the last author is Zhongli Shi.
Right.
And in there is Linfa Wang, who I know. You guys all know. He's a guy who studies bats. Where's he, in Singapore?
No, he's in Australia.
Australia. And Peter Daszak, who I guess is with the EcoHealth Alliance, right?
Right.
Mm-hmm.
And I don't know what he did, but he likes bats also, so he probably did some coordinating.
Yeah, he's probably the coordinator. And then Jonathan Epstein, who, the other one I know on here, he's the fifth author, so he's one of the big bat catcher sampling guys for EcoHealth.
Okay, that's right. There are some Westerners buried in here, but I don't know who they are. Yes. Okay. Anyway, this is a cool study where they've been doing a 12-month survey of a specific kind of bat, Rhinolophus sinicus. Is that the horseshoe bat?
Yes, I believe so.
That's horseshoe bat in Kunming, Yunnan province in China. So for 12 months, they sampled this colony, and they got 117 anal swabs or fecal samples. Matt, how do you get? I know how you would get a fecal sample.
Mm-hmm.
You would get it from the roost, I guess, right?
No, they probably did it the way we did it with our study in Maryland, where you capture the bats as they're leaving for the night to feed using a harp net. If you remember, I put up a picture of that in one of the other podcasts. And then they basically put them in a bag or a container for 5 or 10 minutes, and then as individuals, and then they poop, and you take the fecal sample right out of the container.
But that's not how they describe it in this one. They just talk about putting out 2 by 2 meter plastic sheets under the roosting
sites.
sites.
This one? Maybe I missed that.
They're just collecting it under the sites. And then the anal swab, I can imagine how that's done. For that, you actually have to hold the bat, I guess, right?
You do.
You can't wave the swab up in the air and hope that the bat lands on it, right?
I don't think it works that way.
But have a very low rate of success.
That's right. We had...
So their pooled samples must have been the... for the sheet the mat they put underneath the roost.
Yeah.
Mm-hmm.
And then they did PCR to look for SARS-like viruses, some conserved motifs, and they found that a lot of them, 27 out of 117, were positive by PCR. It's 23%. And they then confirmed those by sequencing. And then they got 2 complete genomes of 2 novel viruses, which they give very nice names like RSSHCO14 and RS3367. Names that just roll off your tongue.
That doesn't make sense to you? Come on.
Can you explain that?
I'm guessing that was just bat number.
So RS is Rhinolophus sinicus.
Right.
SH, I don't know what that one is, but CO, that's the bat number, I guess.
Yeah, I mean, how do you name your strains of polio?
Type 1, 2, and 3.
No,
Just kidding. I'm just being, a little ornery here.
One thing they also did was that they could tell that their fecal samples were from that species of bat because they sequenced DNA from the fecal pellets.
Yeah, that's very cool. This is a common thing now. We heard just this week, oh my gosh, I am so old I can't remember names. Beatrice Hahn was here giving a seminar, and she talked all about this, how they collect fecal material from the forest, and they can identify it as chimp or gorilla by looking at the mitochondrial DNA. Mm-hmm. It's really cool.
Cool.
So Beatrice, in her seminar, she's pointing out all the samples she took, and she turns and she goes, I have a lot of shit samplers in Africa running around for me. I thought that was great.
Mm-hmm.
So the cool thing about these coronavirus, they were able to, from one of these samples, they were able to actually isolate virus. That's from the RS3367. They were actually able to put the sample into cell culture. They used Vero cells, which is African green monkey kidney, right?
Exactly.
Which have the distinction of not making interferon, correct?
Right. So that's why everybody uses them, so most viruses can grow happily in there.
And they got a virus out of here. And it was pretty similar to that RS3367.
Right. So, what we should say is that we had known since 2005 is when the other paper was published saying by actually Lin-Fa Wang, who's on this paper as well, and Dazik. That paper is called Bats Are Natural Reservoirs of SARS-like Coronaviruses. So from 2005, we knew that bats in China had the parent virus to SARS. And so that SARS-like coronavirus, everyone has said that they, is known that's where they originated from, and that virus is the clear parent to the human epidemic strain of SARS and the one that went into animals. The interesting thing about that virus is that at the time that was only sequence, so there was no actual live virus isolated. But when you looked at the spike protein of that SARS-like virus, the older SARS-like virus, it didn't use the human ACE2 protein as an entry receptor. So there's always been this question of no one could really find up until now, this paper, that there was a SARS-like virus in bats that could use the human ACE2 protein. Everyone thought that the virus had to have passage through civets or another animal species in the wet markets and then passed to humans, and that evolution as it went through the other animals was what was needed to evolve it to use the human ACE2. And so what was kind of remarkable about this, one, is that they actually can isolate the virus from the bat samples, which no one had been able to do before. But also that it grows in HeLa cells, which means that it has to be able to use the human ACE2 receptor.
Right. Which they then showed by using cells that have ACE2 or not. And it clearly is binding and utilizing ACE2 for entry.
And it can use the human ACE2, the civet ACE2, and the bat ACE2, all to enter cells.
Right.
So
which
the
is really
civet
the
could have been
remarkable
blameless.
part.
Say that again?
So the civet could have been blameless.
That's right. So you don't need to say it passed through a civet anymore, right?
Well, we don't know. I mean,
of course.
We don't know what the real trajectory was. If you look at the sequence, I'm sure now this paper is coming out, they'll do a lot more phylogenetic analysis of what's known with all the other SARS genomes. But it'd be interesting to see whether you can model the mutation sets as it evolved, if it needs to go through civets or if it can jump directly to human. I would think that you probably need to go through civets, at least as an amplification step, because the amount of people that interact directly with bats is obviously low. So in the wet markets, there's way more people that interact with civets than there are bats.
Right. But that, as you said, may have been simply for amplification and not for selection of something that could utilize ACE2, right?
Right. I mean, what this tells us is that they've identified a virus that is able to use the human ACE2.
Yeah.
Whether that is really the thing that jumped to people in the first place and then maybe went back to civets or around, no one knows.
What they do say here is that the overall nucleotide of sequence identity of these two genomes with human SARS coronavirus is 95%, which is higher than that observed for bat SARS-like coronaviruses in China.
Right.
So this is the most similar, and those were 88 to 92%.
Right. And what this also shows is that SARS is still a virus that is the parent to SARS is still in bats in China and has the capability to infect humans.
Actually, so that's a tough point to say because since this was isolated in the past year, it can't really be the parent, right? Because it's been evolving since the 2000, but presumably some ancestor and the SARS that went into people were in common, right?
Right.
That's what you can say. So this virus has some potential to infect people, right?
Exactly.
Right. In the Associated National Geographic blog that somebody put in the show notes here, one person, Kazek, says he doesn't think it's likely that there would be direct transmission. And he says, if you look at bat-borne diseases, instances of direct transmission to humans aren't common, with the exceptions like Ebola and Marburg. But I was thinking, well, what about rabies?
Yeah, why not? Sure.
Yeah. So
I mean, if there's 3 exceptions, why not?
Yeah.
Why not 4? That doesn't seem to be a good argument to me. They also find that, what do they have? Sera from SARS patients some of them will neutralize the infectivity of this virus, right?
Right.
7 of 9 sera. This is from people who recovered from SARS in 2003. They completely neutralize 100 tissue culture infectious dose 50% of this virus that they recovered. So there's some cross-reactivity as well.
Right. And this virus can infect human bat cell lines, monkey cell lines, and pig cell lines. So it does, it's the real deal. It's really impressive they found this, actually.
I'm surprised they're still looking so hard, I guess. So with EcoHealth and Peter Daszak and Nathan Wolf and John Epstein, I mean, they have a lot of projects around the world where they're doing this. They're looking for novel viruses in animal species all over the world with a focus on bats. At least that's my understanding. So
whether they went in here looking directly for this SARS parent or whether they were doing it anyway and then SARS popped out, I don't know. But I think it's still remarkable that they actually have it.
Yeah. I mean, what was the key? Just persistence? Luck?
Maybe. I think his probably key was funding to go do it.
Right.
Well, what I wondered was, in doing this 1-year survey, did they only sample this one kind of bat? But
Yeah, hard
from
to
the
know.
standpoint of putting out these plastic sheets and getting all this one species, but it seems like they could have sampled more than one kind of bat, maybe? I don't know.
So, Matt, is
I
this...
I was wondering if maybe they were sampling a bunch of different kinds of bats, and this is the one that they found this
Right.
SARS-like virus in. So that's this publication, and maybe we're going to see, in some other kind of bat,
Yeah.
they found something else.
Yeah, I would imagine that this group has a lot of other projects going on at the same time, and that this was kind of a low-hanging fruit that popped out.
This was just the one they got to right off the bat.
Exactly.
Right off the bat. That's a good title. Right off the bat. The Kunming in Yunnan province, Matt, how does that relate to where SARS
So I was looking that
originated?
up. It's not in. It's not really. It's a couple provinces over from where Guangdong is, where SARS emerged. The first cases. But there were cases all over China. I don't see them. I couldn't find a map today of where the most likely where the most cases were.
Yeah. Yeah.
But this would also say that maybe they should go look in people in this area, and I wonder if anybody has antibodies now.
Yeah. They should,
And,
right? That's a good thing to do.
People that were born after SARS, and since then, and you go and look. That'd be really pretty interesting.
Absolutely. So what do you think they would find? Do you think they'd find some sera in people cross-reacting with this?
Absolutely, and this is something NIH should fund all the time.
You going to give up your R1?
I don't know if they'd find any people. Maybe if there were animal handlers in the area and they deal with the bats all the time, maybe you catch it.
Yeah.
Maybe there's other animals in the area, whether it's rodents or civets. It'd be interesting to look at the civet cats that live in the wild around these bats. There's a lot to follow up in. Hopefully they get to do it.
Are you going to get this virus now and work on it?
I have emailed Peter. I haven't got any. I don't know if we're going to get it or not. I don't know if it's not really something directly that we're interested in. I would imagine that they're at least taking the spike or the whole thing and putting it into animals to see if they can get it to replicate. And mice. We have a really nice mouse model for SARS. It's the parent virus for SARS from the epidemic strain grows very well in mice, and we have a mouse-adapted virus that will kill mice with only 6 amino acid changes in the genome. So, all this follow-up is really, I think, interesting to do now.
So the virus you work with currently is it's the sequence of human SARS with some 200 nucleotides stuck in to allow it to use ACE2, correct?
No. So we have a couple different strains. So we have a strain that's called Urbani, which is the human epidemic, one of the human epidemic strains that spread around the world. So that was the one that the CDC basically isolated, or the CDC you can get from the CDC. That virus is there's Urbani, there's TOR2.
Those will grow in human cells, right?
Those will grow in human cells,
okay,
right?
Got it.
We have a mouse-adapted strain, which that virus the Urbani virus, the human epidemic strain of SARS, if you put it into a mouse, it'll grow in a mouse fine. It grows on mouse ACE2 happily. Mm-hmm. It just doesn't kill the mice. Mm-hmm. So if you want to use it, early on, everyone wanted to make a pathogenesis model where you could kill mice with SARS and then look for the viral proteins and then drugs that could reverse the phenotype, reverse the killing. So what was done is different groups, different things, but what was done by Kanta Sabaro and at NIH was she passaged the human isolate of SARS in mice, selecting basically for increased weight loss and disease. Mm. And after 15 passages, in one line of mice, after 15 passages, she had a virus that would kill a Bowsy mouse in 4 days with 10 to the 5th PFU of virus. So, and that virus was sequenced, and there's 6 amino acid changes in the virus out of the 30,000 base pairs. And we did a mapping experiment where we dropped out each of the mutations and put in the wild type amino acid. And when I was in Ralph Barrick's lab, they put that into the infectious clone, and then we did a bunch of mutagenesis on that virus and showed that the spike protein alteration in the mouse-adapted strain, which is called MA15, that spike mutation, which is directly in the receptor binding domain, is what you need. It's essential for the lethality of the virus. But if you just have that one spike mutation alone in the human isolate, you can't kill a mouse. So it's a combination of these 6 different amino acids that's important.
So how do we know that the pathogenesis caused by this selected virus is related to what happens in people?
So that's a very good question. What we know is that the virus infects the same cells as it does in people, that it causes relatively similar lung pathology as it does in people, and relatively similar inflammatory response. So it's a primarily respiratory virus in people as it is in the mouse model. We have 3 different strains of the mouse-adapted virus. So Kanta Sabaro made 2 different strains, one that took 15 passages I told you about, one that took 20 passages that was a separate blind passage. And those have very similar overlapping mutation sets. And then a 3rd strain was produced by Dale Bernard at Utah State University. And that one took 25 passages. And that actually shares a lot of the similar mutations that the other 2 do as well. So the idea being that all 3 of these together show you that, 1, these are the amino acids that are important for adaptation into mice, but 2, the disease, you get this very similar disease between all 3 strains. Got it. So the idea is that correlates with the humans for that level.
So this new virus out of bats that we're talking about, wouldn't really contribute because you'd have to mouse adapt it, most likely, right?
I don't know. We'll have to check. Yeah. Someone will have to check.
I would guess.
What was known before, which was that if you, from the old SARS-like virus from bats, that was 2005, in Ralph Barrick's lab, they synthesized the entire genome. So this is actually, Eric Donaldson was the one that set up the synthesis, and then he was helped by Rachel Graham and a bunch of other people in the lab. And that virus, when you synthesize it, you can make the whole virus genome, you can put it together with the infectious clone, using the same strategy that the Barrick lab has used for a while now. And if you put that into cells, you can get RNA made, but you can't get virus to propagate. So what was needed was that if you take the receptor binding domain of that old SARS-like virus and you swap it with the human SARS spike, so 200 amino acids, I think is what they used, of that spike protein, and put it now into the SARS-like virus, the bat virus, you now can cause the virus grows fine, it'll infect mice, it'll kill mice, it's all happy. So that interaction at the spike level we knew from years ago was really critical for replication and infection. So potentially this new one will actually already be at that step.
Right. So Matt, if SARS reemerged in people, would we be ready with vaccines and antivirals?
It's a very good question. Again, I feel like I'm being led on a line here. It's an interesting question. So over the last 10 years, there has been a lot of work done on SARS. A bunch of labs working on drugs and vaccines and biology of the virus, how it replicates, how it evolves, making pathogenesis models. There are multiple vaccine platforms that have been used and shown to work in animals. None of them have been tested in people, primarily because the epidemic stopped, and so you didn't need to anymore. So I would say we don't have a approved human-tested vaccine for SARS to say this is what you should do for MERS. I think we know from the animal studies what you probably should do if you're going to make a vaccine, and people are doing that for MERS now, multiple different platforms. We're doing a project with the company now. On the antiviral front, we talked about it, I think, when I was the last time I was here. There's a MERS paper out of Heinz Feldman's group and Vincent Munster's group at Rocky Mountain Labs in Hamilton, Montana. And they showed that ribavirin and interferon treatment of monkeys could protect against disease induced by MERS in their model.
Right.
Whether that's really going to be used in people or not, I don't know. Ribavirin has a bunch of other issues with it.
Yep.
But there is...
So does interferon.
Exactly. The really good thing about that study they did was in vitro, in cell culture, they could show they synergize. So you actually use a lot less in vitro, at least. But then at the same time, the Minister of Health in Saudi Arabia, Dr. Memesh, said that they had actually tested ribavirin and interferon already in people during the outbreak in Saudi Arabia of MERS, and they had no effect. And granted, those were on very end-stage cases, so who knows if they would protect early on. Maybe that human study is going to be done. But really what needs to be done is identifying new antivirals that are already FDA approved that potentially could be used off-label for this new virus. And so we're doing that experiment right now. We're collaborating with Lisa Hensley and Peter Jarling at the IRF at it's an NIH lab at Fort Detrick, and we are investigating that right now. So stay tuned for all of those reports. There's other labs doing it as well. There's a lot of labs doing large compound screens to identify lead compounds against the protease or replicase proteins. And we took the idea of doing it against drugs that already are known, what their side effects are known. We know how to dose them, and then trying to test those in vitro against the virus. So at the moment, I would say no. We don't know what to tell people, at least that has been shown in an animal model, other than interferon or ribavirin for MERS.
All right. I want to actually talk a little bit about MERS. Anything else on this paper before we move on?
No. I mean, it's actually a really beautiful paper.
Nice.
I'm really impressed that they got to do it, and I'm really impressed they actually got virus to grow out of the bat samples. If we can get our study going again on bat samples in Maryland, it's something we definitely want to do.
Yeah, so we talked about the 190 nucleotide MERS fragment from a bat a while ago, and I think this is great. You got the full-length genome, and you've got the virus as well. So, I mean, it's clearly replicating in those bats, right? And you had 27% of the bats were PCR positive. So all of those are quite compelling, right?
Absolutely. No, we don't need to look for a smoking gun. This is a good study, and it's clean, and there's a lot of sample to be used and processed, and they found it for real.
Yep. So I guess you're following MERS. What's the latest, Matt? What's going on?
So it's been pretty active last couple weeks, actually, on the MERS front. I think the official CDC count is 150 cases and 64 deaths, if I'm up to date. Just recently, there's been a new case in Spain reported this week, and as well as the UAE in Dubai.
Spain is an imported case, right?
Right. So the Spanish case was a woman who went through Saudi Arabia in the Middle East for 2 weeks during the Hajj, and then came back to Spain, and then she got sick along the way. There's some reports in the media that she was sick while she was there, and didn't want treatment and continued on her way. So I don't know exactly what the story is there, but hopefully we can get some. There'll be virus isolated and sequenced, and we understand kind of where she picked it up and what her travels were inside Saudi Arabia during Hajj. And the Oman case is another new one, and then there's been several recently kind of coming up across the Middle East. So this virus is definitely not gone. It's still spreading, and it's not. The big worry was that during the Hajj, you would have a really large spread of cases, and people would go to Saudi Arabia, go to Mecca, be in tight quarters, and then take the virus back to their countries. And at least on the surface, it doesn't look like it's doing that. But I think that we need a little bit longer to know whether it's really going to spread or not. But the important thing is that no one knows how these people are getting infected, and I think that's really the critical question now. Is what's the reservoir? We know, as you talked about, the paper from Lipkin's group showing that there is the single bat sample with 190 bases. no one has been able to show virus in animals other than that at all.
We did the papers on seropositive camels as well.
Right.
And still there is not a lot of human-to-human transmission except in these settings where you have a hospital with multiple infections. Is that correct?
Correct. Right. It's still a low, very low level transmission. And so I think that the question that we've been dealing with lately, and a lot of people deal with who work on SARS, on MERS, is where are we on this trajectory? So if you think about what we knew for SARS, the first time the rest of the world outside of China knew about SARS, we had 300 cases. The WHO was alerted around case 300 because 30% of the people that were infected in China were healthcare workers, and they wanted to finally alert the WHO and the world that there was this virus going around that they didn't understand, but they knew it was transmitting in hospitals.
How long did it take to get...
Which is not the way to handle the early stage of an outbreak, by the way.
Yeah.
Correct.
How long did it take to get to 300 cases? Did it take a year or less? Do we know?
For SARS?
Yeah.
I don't think we know when case 1 was from any of the papers I've read, actually. It's a good question. I don't know how long it took to get to that point. But I don't think it was... I think there was a lot... There were probably large super spreader events early on that then really grew to make it a problem.
Because with MERS, we're over a year now, right?
Right. September was a year. So we're over a year. We have 150 cases and 64 deaths. So it's still a lot of deaths involved, a high case fatality rate for what we know.
But most of these people have underlying health issues, right?
That's correct. So a lot of them have comorbidity. A lot of them we don't know what that comorbidity is. So we're not getting a lot of information on the clinical side of what's actually going on with the patients, which is problematic for really understanding what the etiology of the virus is. But I think that what it does say is that there has been, I think it was in the Egypt, in one of the recent papers, they actually did look in humans and at a couple hundred sera samples, and they were all negative.
Yeah, you put this link in. It's an emerging infectious diseases paper, right?
Right.
This is in Saudi Arabia. I'm surprised.
Oh, yeah, that's right. It was the Saudi Arabian one.
Yeah.
And so, yeah, that was Stefan Pohlmann's group. So they did. They made a lentivirus with the MERS spike and a couple other coronavirus spikes, and they used those to check for neutralization. And they saw no neutralizing antibody in the several hundred, 158 children and 110 adults from 2010 through 2012. So it's not that, at least in this sample set, it shows that there isn't a very high seroconversion of the population. Whether, how much you need. I think they say that their level of detection, they can basically say it has to be less than 2 or 3% of the population has antibody, even though they got 0 out of 300 or 279 samples. So
there have to be out there some infected people because the virus is not going to just infect really sick people, right?
That's true, right. So what was known, out of the 64 deaths, a lot of the other cases are people that were around the people that were infected.
Yeah.
And so in those people, those cases are called positive by PCR. So they are either BALs or they're throat swabs of patients, or they're close contacts of doctors and hospital workers and family members to look for positive samples. And it's in those that they found these other cases where some of them were symptomatic and some of them were asymptomatic. So that's kind of the worry is to know whether a asymptomatic person can really share the virus or not.
Yeah. I was looking around in the literature, and there's a couple of papers I wanted to mention, and one of them is yours. You guys just published a paper on MERS in mice, right, Matt?
We did, a long time coming, but we just basically did a basic study very early on, as soon as we got the virus last year, to check in both wild type and innate immune deficient mice, STAT1 knockout mice, and STAT1s we used for SARS to show that they actually have more severe disease for SARS than they do for wild type mice. So we tested all those strains, and we basically couldn't find any virus. So MERS doesn't replicate in mice, and other groups have communicated with me. They actually have the same result as well.
So, but I wondered whether you tried any of the wilder outbred or wilder inbred strains, or even outbred strains.
So we haven't done it here. I've heard from other labs they have done that, and they also don't see any infection replication. I can guarantee you if someone had a mouse that was working at the moment from one of these outbred strains, we would see a very beautiful science paper describing it, I would imagine.
Right.
So at the moment, we don't see, we're not seeing that the mouse model is working in the current state. So I think that means, though, is that we need a transgenic mouse making the human receptor. And there's multiple groups trying to work on that now.
But I have to say, kudos for publishing people would consider a negative result.
Absolutely, yeah.
Yeah, you'd be surprised how many people didn't like seeing this paper.
Yeah.
So we had a little bit of an
issue.
This is
hugely important that people, you published this, and now people know several places not to look.
Well,
right? It saves a lot of duplication of effort.
I mean, I've shared this data with the people in the field, and other people in the field have shared their data with us. So, I mean, we knew this data for a while, and I've been sharing it when I give talks about MERS. So, this wasn't something we were keeping to ourselves, obviously, but
to have it in the literature, I think is really, really important, and I think I just wanted to commend that because there's too little of that going on.
For sure. Mm-hmm. So the receptor is dipeptidyl peptidase 4, and you mentioned in the paper that you think the low level of mouse DPP4 in the lungs is what makes them not susceptible.
Yeah, so I think it can be both. It can be. I mean, we don't see a lot of it there. Other groups have seen some DP4 in the lungs, depending on if they look at RNA or protein. So I really think that if you look at if you map the interacting domains of where the DPP4 interacts with spike, there are mutations in the mouse, the mouse receptor that make it seem like there's reasons why it doesn't work with the human, with the MERS. So I don't think that that's really. I think expression can be one reason, but I think that biology is another reason, and that interacting domain is important, clearly.
So you guys aren't making it transgenic, are you? Actually, you might not want to say that, but it's a good thing to do, right?
It's the next thing to do. We'll put it that way.
You said several groups. I'm sure lots of people are doing it, right?
Yeah, there's other ways of doing it. I mean, the question is, do you need it in the certain cell type where the human protein should be expressed? Can you put it all over? Can you make a just constitutively expressing receptor?
Yeah.
Is it Clara cells? Is it type 2 alveolar cells? Is it is there another cell type in people that this is really important for? I think one of the remarkable things about MERS is that there is not a single human lung sample showing disease of an infected person with MERS. We don't have an H&E stained lung that's of a MERS infected person to see where the virus is. So I think that I think all of those kind of human clinical samples are really needed to really understand what's going on
Why is that?
Well, it's, most of the cases are in Muslim people, and there's a lack of autopsies in those patients.
Autopsy, right?
So that's really the reason. And of the people that actually have not died, some of them are not Muslim, but they're obviously not dead, so they're not going to give us their lungs.
Right. Yeah.
So we still we need those clinical samples really to understand what's going on.
Right.
And that's really the next. Right. That's one of the next steps in the clinical side to really understand how this virus is doing what it's doing.
I've heard that from a number of people, that the lack of clinical specimens has made it difficult to make a lot of progress here.
Yeah, I mean, I think that the Saudi government is trying to do as much as they can and to release as much information as they can. I realize there's a privacy issue, and they don't want to say exactly the people's names that are infected, what they died from, what other comorbidities they had. I mean, there's other things involved when there's HIPAA issues here, but all the information would be important for really understanding the virus. So
sure.
Hopefully it'll eventually all come out, and we'll be all on the right track.
It's very curious that it is really, the infections are really limited to that area, right? It's just not going anywhere else. I mean, people are bringing the infection elsewhere, but that has not resulted in it being anywhere else.
Well, there's the one case in Egypt now, and the one case in Oman.
Yeah.
Both places where they've done the camel surveys, where they show that they're all, most of the camels are seropositive. So now at least we have a connection there between an animal and a person and people being infected. Whether it's really from camels or not, we don't know.
Yeah.
No virus has been isolated from the camels at all.
Right.
So, all of these things. I mean, I don't know if anybody listening to this doesn't get excited about MERS, who is a virologist. They're crazy, because this is kind of what you live for as a virologist, and we're really having. It's really an amazingly interesting time right now to do this stuff.
It's also frustrating, too, because you can't get what you really need to sort it out.
Well, I mean, it's true. On one level, that's true. I mean, we do have a lot to do outside of just getting the clinical samples, so I'm fine with what we're doing. We're busy enough.
You have virus, so you're a virologist, and you have plenty to do, but it may not sort out what is the source of the infection, right? For that, you need to be there, get samples, and so forth. So
right.
I'm sure
it's all being worked on.
Yeah, I'm sure it'll come out eventually. I like this other paper where I found where they looked in and found a very related virus to MERS in European hedgehogs.
It's pretty good, right?
Why would you look in hedgehogs, Matt?
So I asked that question to one of the authors.
They are cute.
This is another paper from Kristen Drossen's lab and Jan Drexler, who have done a lot of virus hunting work in the past, especially for coronas. And so I asked Marcel Mueller, who's one of the authors, why they looked, and he said they were looking for other animals that ate insects, similar to bats, and to see if they could find similar viruses like they were finding in bats. The Drosten lab and the Drexel lab have done a lot of bat virus hunting, and Marcel is kind of the key person in a lot of their studies, and so they were really looking for other animals that ate insects, and so they happened to come across this hedgehog. What do they call it? A hedgehog animal shelter, which was taking in sick and injured hedgehogs.
It's a prickly business.
I guess those exist.
It's a prickly business.
That's a good title.
It is. I'm going to write it down here.
And so they went to the hedgehogs and they looked to see what's there.
Are hedgehogs also close to bats in some way? Is that correct, phylogenetically?
I don't. Kathy, you put this up?
So, my interpretation was that there are things closer to hedgehogs closer to bats than hedgehogs, depending on which phylogenetic tree you look at. But I mean, it's neighboring, but there are other things closer.
Yeah, in the paper they say the next sister taxon to bats is this hedgehog.
But these phylogenies suggest otherwise.
Yeah, it's weird because in the abstract of the paper, that's what they say.
Yeah, I was confused by that too, especially when I saw these trees that Kathy put here. It looks to me like I don't know, to pick a couple of random samples, camels and civets and other cats would be.
Right, and in another one that I think I didn't paste in, I decided that maybe rhinos, horses, and tapirs would be closer, but. I suppose that there's just as many ways to do these phylogenies as there are animals.
Well,
if you ask three taxonomists, you get 5 answers, I'm sure.
Well, this phylogenetic tree that you put up, Kathy, if you look at the bottom, you have the Chiroptera, which is on one branch of a branch, and then there are many other sub-branches on the other end, but then the Eulipophilia, which are the hedgehogs, are actually the other branch from which the Chiroptera are coming off. So in that sense, it is the sister taxon.
Sure,
right?
sure.
So maybe that's that. But yeah, you would think these others, Carnivora, Pholidota, are all closer to Chiroptera than...
Yeah, but I guess there are fewer branch points.
Exactly.
I guess the taxonomic distance to...
is fewer, is less.
Eulipophilia is shorter.
You also just, you take what you get. If they look for things around them that you can sample, and if it's hedgehogs, you say, oh, yes, please.
So Matt, they basically found a corona highly related to MERS coronavirus. Is that right?
Yes. It's in the same general family tree as MERS. If you look in the phylogeny in this paper, you can see it's definitely an offshoot. It's not the next neighbor,
right?
But it's in the same clade. It's a clade C coronavirus, so it's right in there.
And 60% of their specimens were positive for this.
Yeah, with very high viral load, which
Yeah.
is really remarkable. So, I mean, what this shows us, and also the sera will light up cells infected with MERS as well. So it does cross-react to some level. And I mean, and what it shows you, there's way more out there than we knew, right? At the very beginning, we talked of the podcast, we talked about how technology changes science. And I mean, if there's anything in the last 10 years that we've learned, it's that by deep sequencing and using these, looking out there again to see what's out there by PCR and new sequencing methods, we can find stuff we never knew was there.
Are there hedgehogs in Saudi Arabia?
I'm sure there's some relative of hedgehog there. I'm sure.
I'm surprised you don't have the answer on the tip of your tongue.
No, I didn't think about looking for that. I do have a slide in one of my talks where I show all the animals that I found online in Saudi Arabia, and I'm trying to see if hedgehogs.
Desert hedgehog is.
There's a desert hedgehog. There you go.
In the Middle East and North Africa.
Yeah, perfect.
Oh, they should go look in there. I mean, it could be important, right? Obviously,
Absolutely.
It's neat.
That one's not as cute, at least in the Wikipedia picture. It's not.
Yeah, they live. Strong, consistent number all over. Saudi Arabia, Israel, Egypt, Ethiopia, Algeria, UAE. I mean, every place. There you go. Yeah. The hedgehogs are the key to MERS.
So it could be that the hedgehog virus was the source of the human infections. Is that possible? According to the phylogeny.
I mean, who knows?
Because we don't have anything except for 190 bases. We have nothing from bats, right?
Right.
We have some more distantly related viruses in some European bats, I think.
Right. So the same group, the same group that did hedgehog work, or at least some of the people, are the ones that did the European bat, isolating, seeing a MERS-like virus there, and then they have African bat samples as well. So, I mean, I think what it shows us is that there are viruses that look like MERS all over the place,
right?
Or at least similar relatives of MERS. Whether and why those don't jump, we don't know. And why why Saudi Arabia seems to be a hot a focus of this, we don't know.
MERS don't jump.
There you go.
We have a thing in our textbook called Enzymes Don't Jump, based on the same basketball thing. MERS don't jump. I can't do that. I couldn't use that in the title because it's just grammatically wrong, but it doesn't jump. It's pretty cool. All right. Then there was another paper I stumbled across, which is also cool because it uses an approach that we've talked about before on TWiV, and that is looking for host pathogen interaction by looking for the ratio of synonymous to non-synonymous changes. And this is from Eddie Holmes. And Lin-Fa Wang, and what they did is they compared sequences of the receptor dipeptidylpeptidase 4 in bats and also other various insorted species like sheep and killer whale, and they found evidence that there's been some adaptive pressure on specific residues of bat DPP4 compared to the other species. So these are all sequences that are in the database. You can just go through and pull out the DPP4 and say, are there any residues that look like they've been positively selected, which would suggest that they're changing in response to a pathogen putting pressure. And since we know DPP4 is the receptor for MERS, that might provide some insight. So they found that 3 of these 6 amino acids are in fact located at the interface between DPP4 and the coronavirus spike protein. And that crystal structure of the 2 was just solved recently, which is another paper out there, which is really amazing. It's really, I mean, that's a big deal to solve the receptor spike protein together.
Right. I mean, what all of these papers, outside of just the basic data show is that everything that we have learned for SARS in the past 10 years since SARS emerged, it's completely set us up to understand what's going on with MERS. Right. It's really a beautiful story at that level. All the funding that NIH gave and all the work that's been published on SARS in the past, everyone is shifting over to MERS and doing. Using their specialties to figure out what's going on here. So it's really a beautiful collaboration at that level.
So this paper would suggest that MERS coronavirus has been putting some pressure on bat DPP4 over the years, which would suggest that the virus is in fact in bats, which of course we haven't seen yet. So that's pretty neat. It's a pretty short paper, and I would have liked them to do some mutagenesis and look at this specifically, but I guess that's for the next paper as well. It also would be interesting to do this for bats that are in the Middle East. So they just picked bats whose sequences were available, and I think the ones that they looked at were not actually bats from the region. Vampire bat and Pipistrellus. I don't think those are the bats in Saudi Arabia that have been implicated, but it's quite interesting anyway.
Yeah, absolutely. I think that hopefully the next step is going to be biology of this to show you can mutagenize the spikes and you can mutagenize the DP4s,
right? And
you can now show that interaction in a cell.
Yeah, you want to show that these positively selected residues have an effect on binding, right?
Right.
Because all we know is that they're in the interface of the structure, which is suggestive, but it doesn't prove it. And that's, of course, what Sarah Sawyer did with the transferrin serving as a receptor for a variety of viruses. So I'm sure that's next.
Right.
Anything else, Matt? No, I think that's it. I think we, at this stage for MERS, I think everyone is working on it, and there's a lot of people pushing ahead on therapies and biology and drugs and animal models. And so I think that's going to be the next step of, series of papers coming out from all these groups.
All right. Well, we'll be here to cover it. It's our job. Let's do some email. Kathy, can you take the first one?
Sure. Mark writes, "Hi, TWiVers. I enjoyed your email episode number 252. Regarding DJ's letter on anti-vaccination sentiment and your panel's discussion, here is a video supporting vaccination." He gives a YouTube link. It is a slightly mildly satiric parody of the anti-vax movement. Subtitles in the last few scenes. All the best. And it's been a while since I looked at this, but it is kind of good satire. I quoted a line, "Use of vaccines without birth control can lead to explosive population growth." And Rich had commented on this one too. It says he liked, "Do not take vaccines if you've chosen to die from the mumps." So it's
cool.
It could be useful.
Yeah.
You put a couple of links in here too, Matt, right?
Yeah, the CDC has a page on myths associated with vaccines, and that's worth reading.
Right.
And then, just go. We have covered it before, but there are measles and mumps outbreaks around the country now linked to people who don't get vaccinated. And herd immunity is very important for all of these, basically the elimination of all these viruses, since everybody doesn't have a super high titer of it. But if enough people around you do, then you're protected. And when somebody comes into your location and your vaccine titers are low, and they're positive, then you can get infected, and that is why people get spreads.
Yep. Matt, can you take the next one from Cassie?
Sure. Hello, Professors TWiV. I know you were all always on the hunt for science-related geekery, and I don't know if anyone had run across this before. And he has a link to, or she has a link to this nitoshop.com. I just bought the Pandemic Risk shirt for myself, and it's super soft and comfy. And she comments that it's 88 degrees Fahrenheit, 32 degrees Celsius with 60% humidity here in Austin, Texas. The end of a beautiful, clear, sunny day. Thanks for all you do.
Cassie, we were just in Austin. You didn't come see us. Or maybe you did. I don't know.
Maybe
Anyways,
she's shy. Yeah, but she probably wasn't wearing her shirt. You would have recognized her.
I would have. It's a cool shirt. Yeah.
Yeah. It's a cool site. Good stuff.
Yeah, it is nice. Alan, you're next.
Okay. So Peter writes, "Hi TWivers, I would like to commend to you episode 101 of the Brain Science podcast by Dr. Virginia Campbell, Synapse Evolution with Seth Grant." It gives a link. I don't know where to submit this as a listener pick, as the connection is at the ribosome level. It could be in any of the TWiCs, but like weather sites, it's just cool. By the way, beautiful spring night here in Sydney, Australia, 19 Celsius at 10 p.m. and calm. This Brain Science podcast episode is the best single podcast episode I have ever experienced, and I am a podcast listener. So what's so special about this podcast? 1, a full and satisfying explanation of where our brains came from in evolution, starting with the first multi-celled organisms, to the ribosome level. 2, it turns out, maybe probably mental illness is the price we pay to have evolution. 3, a lot more. Vincent, when I first saw you on video, I was shocked. You look nothing like you sound. But I like your shtick. I watched quite a bit of the live episode 250 with Robert Garcia and was very impressed. You're so natural. Back to the point. I think this is one podcast episode every citizen of planet Earth should listen to. It's about more than the subject matter. It's about what is possible. I will try to record this as an MP3 tomorrow and send it in as well. It's just that the link would need to be in the show notes, as it would regardless, I suppose.
He was shocked. I don't know what to make of that.
You know, about this, we always say that the person doesn't look anything like we thought they did, just from their voice. But I've tried to think about the various NPR commentators or reporters that I don't know what they look like, and I don't have any picture in my mind for them. I think it's just then getting their face associated with their sound is remarkable. At least for me, I don't have any preconceived idea of what they look like. It's just it's blank, and now I have something associated with it.
So that's shocking.
I'm the same way. When I've heard somebody on the radio, it doesn't matter how long I've listened to them, I really don't have a picture of how they look.
Yeah.
And Vincent, I would never have guessed you had a beard.
Really?
Never. Your voice, you just can't hear it.
Yeah. You're right.
He keeps it away from the mic.
That's pretty funny.
Well, the first time I met Rich Condit, I was standing in line at ASV, and I heard this voice. And I was like, hold on, I know that from somewhere. And then I turned around, and it's him. So it was very funny to match that voice to a name.
I find myself trying to imagine what people do look like from their voice. I do that a lot for some reason, and then it's always totally different from what I imagine. And so I think I stereotype voices with people, and that's what I get surprised about. Anyway, this Brain Science podcast is very good. It was a pick a long time ago on TWiV, and you should check it out. She does a good job. Oh, I get Robin's. Okay, Robin writes.
Now, Robin generally writes about something that we said on an episode, but doesn't reference what episode it was, so it's sometimes a bit confusing.
Yeah, and there are usually multiple things. So
yes.
All right, Homo calidus possi sapiens ? Dumbest people mess up the world the most. Actually, the ones who mess up the world the most are the ones who are smart, but not wise enough on how to use those smarts. Homo calidus possi sapiens. I guess we talked about this on some episode.
I think it was the one about the dinosaurs were the dumbest, and then somebody else wrote in to you about that. And I just parsed out what Homo calidus pausa sapiens would be. The calidus is warm, hot, fiery, fierce, and the pausa sapiens would be few or little wise. So,
yeah, I guess so. All right, continuing. Humanity has successively defeated each limit to increasing human biomass through technological innovations accumulated through cultural transmission, from the first modification of sticks and stones to the principles of virology, allowing an overshoot of the population well beyond the carrying capacity, absent fossil fuels. All right, so I guess we talked about why dinosaurs got so big, and this is possibly explaining why we didn't. Thermodynamic footprints. Does this link have to do with thermodynamics?
Yeah, he gives a link to that.
Measuring human impact.
Right. Gives a link to thermodynamic footprint. This has also been discussed in Too Smart for Our Own Good, The Ecological Predicament of Humankind by Craig Dilworth, who also has a free online synopsis. Overpopulation and the Vicious Circle Principle. This is an article by Craig Dilworth. That's what he just referenced. And finally, podcasts for Christopher, the physical therapist in Idaho. He gives a link to podcasts about physical therapy. PT podcasts, or PTJ podcast. There you go. Because I guess Christopher was saying his wife thought it was weird that he listens to TWiV instead of stuff about what he works on. So, Christopher, you cannot give up TWiV for this podcast, but you can do both. All right. All right, Kathy, you are next.
Okay. Sam writes, "Hi, Twivers. I'm finally picked up your podcast. I've been fascinated by science all my life, so I'm familiar with most of the subjects you talk about. But as a non-virologist, I do find the discussion going over my head on a regular basis, and that's great. I learn new things about the astounding world of life every time I listen to TWiV, TWIP, or TWIM. I write to you from Tucson, Arizona, where it is now 89 degrees with 11% humidity. A lovely fall day with a touch of chill in the air by desert standards." My email is prompted by an article I am reading, and he gives a link, which discusses the relevance of domesticated viral genomes, which must have been in the ancient past, to cancer. Some podcasts ago, you talked about the association between bacteriophage viruses and mucus, with the paper under discussion finding that mucus was well adapted to host phage virus as an effective defense against infecting bacteria. I wondered at the time, could it be that these phage viruses aren't environmental in origin, but are expressed by animal cells? It seemed implausible that a virus that infects bacteria would end up as part of an animal germ cell line. But in the fullness of time, I suppose it might happen. And if it did, it would give its descendants a decisive advantage versus bacteria. What are your thoughts? Thanks again for the awesome podcast, Sam. So it looks like Matt looked into this a little bit.
Yeah, so there's a professor here named at University of Maryland with me named Julie Hotop, and she works on lateral gene transfer between organisms, specifically like bacteria and Drosophila and bacteria into people. And so I asked her about this, and she actually has a paper which kind of references this for cancer genomes, where they see bacterial chromosome pieces that are integrated actually into cancer genomes in people. So we can put the link in the show notes if you want. But she said that she thinks that, so from her email, she said basically that some people would say that human cells would die after you get integration of the phage, and maybe not had enough time for the phage particles to be made. But potentially, if the phage actually integrates into the right stem cell, you could get propagation and act as a basic early primitive immune response, where you get phage production and then you get clearance of the bacteria that the phage would normally infect in your gut. So I think it's an interesting idea. I don't see why that couldn't happen. I mean, I think there's obvious genome issues of why it would be hard, but we have a lot of transposons and other jumping and movable elements in our human genome that I'm sure jump relatively frequently.
Yeah, it's an interesting idea. There's no evidence that cells could make phages, right? But there could be pieces of them in it.
Right.
I saw this paper of hers in PLOS Computational Biology. It's really interesting. I think we're going to do it on TWIM.
Oh, good. You should call her. She's really good.
Yeah? Okay. Maybe we can have her come on and talk about it. But it's an interesting idea, Sam. Thanks for that link.
But again, it's a great question from someone who doesn't do virology at all. I think it's great.
Yeah. The paper that Sam originally linked to is very interesting in itself, and we should talk about it at some point. It's called VLINC. That's very long intervening sequence non-coding or interspersed non-coding RNAs controlled by retroviral elements are a hallmark of pluripotency in cancer. So basically, these are a large part of our genome is non-coding, and a lot of it is transcribed into RNAs. And apparently, the endogenous retrovirus sequences that we have supply the promoters for a lot of these RNAs to be made. And their presence, the amount of these RNAs appears to correlate with pluripotency, that is the ability to give rise to different kinds of cells and cancer. So they actually even knocked them down in a cell line, and it made it less malignant. So it's really interesting stuff. Non-coding RNAs. Matt, I believe you are next.
David, okay. Dear Vincent, Kathy, Dixon, Alan, and Rich. I guess I'm not included.
Sorry, Matt.
It's okay. I started listening to TWiV about 3 months ago in anticipation of my retirement next July. Finally, in parentheses, finally driven by the lack of funding from the bench where I've continued to work. And my initial sense that TWiV is even better than the journal clubs we have around here. And so this is David Spector at Penn State. He says, I've not been disappointed. The science is great, whether virology or other. Just as important, the interplay among you models the kinds of relationships that should serve to help recruit young listeners who may mistakenly think that there is not enough social in scientific research and scientific career. On TWiV 253, Vincent said that the oral polio vaccine is delivered in 3 doses, but only one serotype will establish infection and confer protection at a time. I learned that multiple doses are given in case the recipient has an active heterologous viral infection of the gut, as that too could prevent a take. Clearly, both these things cannot be true, because if they were, the frequency of vaccine failure against at least one type would be significant. Or maybe this kind of failure is common, and we don't notice because the vaccine strains circulate in a vaccinated population, and so one might just acquire the missing immunity a bit later upon environmental exposure. Please comment. And he comments that it's 14 degrees Celsius and 57 degrees Fahrenheit with overcast skies and light rain here in Chocolate Town, which is Hershey, Pennsylvania. Please, special shout-outs to Kathy, who I've known for many years, many more years than she may want to acknowledge, and to Vincent, who visited us not too long ago. Keep up the good work, David.
I guess you know Dave, Kathy, right?
Yep, from the adenovirus. From quite a long time ago.
And he's just retired, apparently.
Well, he's going to, it says. Yeah, and he's been continued to work at the bench, so till next July at least.
Yeah, 3 months.
Good.
Well, I asked Mark Palanch, because he's the polio vaccine guy at CDC, and I said, why do we give 3 doses? Because each one only one takes each time. And he said, the rationale is, as you stated, the more doses, the better. For IPV, inactivated polio vaccine, it is actually the absence of enough convincing data that 2 doses are adequate for protection in a high enough percentage of the population, clearly is 90+%, whereas 3 doses is 99% in the U.S. and Europe. The second IPV issue is the need for a booster, i.e. a 3rd or 4th dose separated from the primary doses by a period of at least 6 months. All of these uncertainties of number of doses and timing has led to a plethora of schedules of routine IPV use globally. And he sent a paper which is so scary. It's a decision tree paper on how to figure out what kinds of polio vaccines to give people. He says, no optimal approach is in sight. It's I don't know if you've looked at this paper, but it is so scary. It's full of all these complicated trees. So there you go. So the idea of gut inhibition, David, that was used for a while to explain the poor take in India, where I think there are many, many more other kinds of infections than we might have here. I'm not sure that's true or not. So, I don't really know what else might explain it. And I agree with you that, here in the U.S., we certainly have a lot of things in our guts as well, yet we do pretty well with polio takes. And I don't think it's because the vaccine strains circulate in a population. I think it is the vaccine itself, but we just don't know.
So, Vincent, what do you think about the polio stories that are coming out now from the new infections?
Well, in Syria, there's a big outbreak of at least 20 cases. And the cause of that is quite clear. The immunization rate went from 90+% to 46% because of the civil war. So it shows you that if you don't immunize against polio, you will get polio, because as long as there is wild virus circulating, and it still is circulating in Pakistan, Afghanistan, Nigeria, it spreads elsewhere. There are several new countries in Africa that have had outbreaks this year compared to last year. In every case, it's because immunization coverage drops. So it's a really tough problem. You have to cover everything. As long as Nigeria, Pakistan, and Afghanistan continue to not be able to get rid of wild polio, this is going to be the situation. It's very tough.
Is there anything about the strains that are emerging now that makes them special or different than what was known before?
They're type 1 polio. It's the same, more or less, that's circulating in those other 3 countries. There's nothing new about them. It's just a drop in immunization coverage. Would you agree, Alan?
Yeah. Yeah, it's the same. I mean, it's the same virus. We knew it was there. And you stop vaccinating and it shows up again, or you get vaccine-derived.
Right.
One or the other.
All right, let's just do 2 more here. Alan, I think you're up next with Stephen.
All right. Stephen writes, this Nature News piece includes a nice explanation of natural RNAi, naturally occurring RNAi mechanism, and links to a Nature News piece that's open access, by the way. You can read through this explanation of how the RNAi system works, and there's a nice video to go with it.
Yeah, this is good, since we just had 2 episodes on RNAi. So if you want to learn more, you can do that. And the last one is from Todd. Hi, gang. After the high praise given to this book by many of the TWivers, I downloaded the audiobook and listened to it over a lengthy daily commute I had to endure last fall. I loved it. Now it's soon to be a 6-part miniseries by Ken Burns on PBS. And he's referring to The Emperor of All Maladies. That's cool. It's a great book. Cheers, Todd in northern Colorado. I hope they do a good job.
Yeah.
Right? They could mess it up. It's a documentary. So I guess they're going to sort of tell the story in these episodes. So we'll see. We'll check it out. It's a great book, though. I just heard him talk here not too long ago. All right. Let's do some picks of the week. Hey, Matt, what do you have for us?
So I asked permission, and I guess I'm allowed to plug this book. My brother is actually a writer on the side, and he has a new book out, which I will plug. It's called Reply All and Other Ways to Tank Your Career, a guide to workplace etiquette. His name is Richie Frieman, and he is actually the modern manners guy on the web. He has a website and through quickanddirtytips.com. So if anyone wants to, the funny thing about the book is he did there's a bunch of cartoons in it. He did all the cartoons. He did all the writing, and it actually works really well for lab etiquette as well, I will say, not just business etiquette. So you should go link it, buy it, support the Frieman family.
So he's the successful one, huh?
Yes, exactly. My mom got her Jewish doctor, and then my brother was the artistic one. So if you look in the PLOS pathogens, the PLOS pearls that I wrote on MERS, we have the cover image. If you could have a cover image for an online journal, but if you go to PLOS pathogens, where the issue from a couple months ago, where we have our MERS paper, my brother painted an oil painting of virions, and so he has the cover image on there.
Oh, cool.
So he's helped us out with a lot of pictures for the lab. So
we'll have to put a link to that article as part of the show notes. And since PLOS is open access, everyone will be able to read it.
There you go.
And we could use the image too if we want.
Sure. So, and if anyone would like other pictures of viruses or various bacteria for your lab or your university, please get in touch with me. My brother will be taking
commissions.
He'll be taking options for and put them in his queue for sale. So Let me know.
Hey, that's pretty cool. I didn't know he did that stuff. Pearls MERS article.
When searching for the book, I found a link to an audiobook sample, and I guess it's him reading it.
That's him.
Okay, and there's also a chance to win a free copy. So you could even go check that out.
Cool.
Get yourself
Absolutely.
a free copy of it.
But Matt, you can't enter.
I cannot enter.
No, it's not fair.
But yeah, go. My brother's good. He's very funny. He's a really good writer, great artist. So support the Frieman clan.
Absolutely. Is he in this part of the country or somewhere far away?
Yeah, he lives in Baltimore, right around the corner from us.
Oh, so you must see him.
Yeah, so we see him all the time. He's great. I love my brother.
Very good. That's great. Kathy, what do you have?
Well, in contrast to the YouTube video I picked a couple of weeks ago for John Holland that was almost an hour long, this is the other extreme. It's only 24 seconds long. It's an optical illusion. And then once you're on that YouTube page, you can click and see lots of other kind of optical illusions. And I just thought it was fun. It's very short. And even if you go down the rabbit hole of watching several other YouTubes, they're all short too. So.
This is fun.
I'm watching it now. It's great. It's great. It's really good. Really nice. And,
It's fake.
It's silent, so it's not a problem here. Alan, what do you have?
I have a blog that I stumbled on recently. It's called Amboceptor. It's actually, I think it just started recently, and this blogger followed me on Twitter, and I saw the link to the site and started reading, and I've been hooked ever since. It's now on my RSS feeds. I don't know who is who's behind this, but Amboceptor is someone who's very interested in immunology, microbiology, public health, and history. And so he or she digs into the historical literature, often from the could be from the mid-20th century, could be from the early 20th century. I think there's some stuff from the 19th century in there as well, and looks at the kind of where these odd little corners of scientific discoveries come from.
Very cool. It's Mike, Guy.
Mike, yeah, okay.
It's very nice.
I did know of a woman named Michael, but
You
it
did?
probably is a guy. Yeah.
Right.
Yeah,
The posts aren't
that's
a bit-
true. I know women named Alex, too. Yeah. All right. I stand chastised and corrected. Okay. Just like Robin could be a man or a woman, but I do happen to know that the Robin who writes us is a guy, because when I get his email on Gmail, it's got a picture of him.
Ah, yes.
That leaves me. I have a video from the American Society for Microbiology called The Microbiology of Beer. And this is a video of a colloquium, which was the ASM, and in particular the American Academy of Microbiology, which is part of ASM. They sponsor these colloquia at headquarters in Washington, D.C., where they get a couple of scientists to come and give talks, and they invite the public to come in. They can fit about 50 people into the room, and they live stream it, and then they make a video. So my friends over in the communications department do the streaming and the video. So this is a cool one on beer, and they have a guy, Charles Bamforth, who is very entertaining and good, and then Rebecca Newman, who is from a brewery called the Dogfish Head Craft Brewery. And
Excellent brewery, I might add.
yes, and they talk about how you make beer, and it's a lot of fun. And if you happen to be in the D.C. area, they announce these ahead of time. You're welcome to go to them. It's free, and in this case, they actually served beer. They are allowed to serve alcohol at the headquarters of ASM. I know that for sure because I've had it there before. Anyway, the video is cool, so check that out. And we had a couple of listener picks. You may have heard one from Peter, I believe, and then there was another one before that, which is escaping me, but they'll all be in the show notes. So that's the Brain Science podcast from Peter. And there was one more, but I don't remember what it is.
There was the vaccine video.
Vaccine video. We'll put those all in the show notes, and that will do it for TWiV 258, which is notable because it's the 8th appearance of Matt Frieman on our show.
If I get to 10, do I get a free R01 or something?
There's no such thing as a free R01, but I'll give you a beer. How's that?
That sounds good.
You're like the
catch you
Alec
at the next
Baldwin
ASV.
of TWiV.
The Alec Baldwin of TWiV?
Yeah, I think he's hosted SNL more than anybody else.
Ah,
Yeah,
okay.
Either Alec Baldwin or Steve
Yeah.
Martin.
Matt, I'm sorry that we can't make you that famous.
That's all right.
but
I'll take what I can get.
Among scientists, it's pretty darn good. Anyway, thanks, Matt, for joining us today.
My pleasure. It was great. Thank you.
Matt Frieman is at the University of Maryland in Baltimore, or Baltimore, however you want to say it. Balmer. And you were also born and raised there.
I was.
Cool. Kathy Spindler is at the University of Michigan in Ann Arbor. Thank you, Kathy.
Thanks. This was fun, and I learned a lot.
We do. Alan Dove is at turbidplaque.com and also on Twitter. Thank you, Alan.
Always a pleasure.
And I'm Vincent Racaniello. You can find me at virology.ws. You've been listening to This Week in Virology. Thanks for joining us. We'll be back next week. Another TWiV is viral.